Okay, so I hope it's going to work. Anyway, thank you very much to the organizing committee of this conference for the privilege to speak here. And today I'd like to share with you some of our results on psychedelic drugs and the safety problems related to their use during pregnancy. Psychedelics are compounds that are able of altering consciousness by acting on serotonin receptors in the brain. And the term psychedelic was coined from Greek for the first time by Osmond in 1956. Psychedelics are molecules that are derived either from plants or from fungi, usually, or less commonly but with increasing sort of popularity, they are synthetic, of course. Psychedelic substances have been used by humans for at least 10,000 years, mainly by certain tribes in their religious rituals. But it was first suggested in the 1980s by Glennon that the target of their activity is the 5-HT2 receptors. And he demonstrated, the group demonstrated this on a rat model. Okay. Research on psychedelic compounds was kind of popular until the 1970s, but then as they became illegal under the Controlled Substances Act in the USA, for example, that was passed in 1970 during the Nixon administration, the research progress in this area became relatively slow because the government agencies did not want to give funding for clinical research on such compounds very easily. And on the other hand, some researchers preferred to play it safe, and they avoided topics requiring physical access to the samples of controversial or prohibited substances. But within the last 15 years, the situation has begun to change, and the research papers and grants in this area began to surge. It looks like that in present, the studies on psychedelics focus mainly on the possible new applications of such compounds or their structural analogs in pharmacotherapy of pain, addictions, and some psychiatric disorders, like, for example, drug-resistant depression or anxieties. And also the structural modifications of known psychedelic molecules are designed to not to give the compounds that could be used for such applications, but on the other hand, without the unwanted kind of activity in the central nervous system, and also with the improved safety profile. But it's kind of surprising, perhaps, relatively little attention is being paid to psychedelics and their analogs in the context of pregnancy. The safety of pregnancy, as far as such compounds are concerned, is to a large extent unknown at the moment, because obviously for ethical reasons, no research on real pregnant women might be conducted. And as a consequence, the majority of what we know on the impact of maternal ingestion of psychedelics on fetal development in humans comes from case reports, which are quite scarce, and retrospective survey studies, which are not very reliable because these are usually studies on random groups of subjects in random conditions and without proper control groups. And according to some studies, the majority of women that participated in such studies had unplanned pregnancies. Many of them smoked cigarettes, many of them drank alcohol, and a number of them also used multiple drugs. So it was very difficult to tell which ones the isolated effects of psychedelics on the unborn children are. Interestingly, I would also like to mention certain results published quite recently, but as it was demonstrated on a rat model, in some situations, neurodevelopmental issues in offsprings are not directly linked to biochemical, neurobiochemical effects of the psychedelics during pregnancy, but more may be attributed to the lack of proper maternal care by drug-addicted women, basically. Anyway, from those very few clinical studies that were conducted, the majority was conducted on LSD, and it was found that some children born to mothers that used LSD during pregnancy suffered from abnormalities such as finger webbing, missing or underdeveloped limbs, tibial and fibular aplasia. But on the other hand, there are also there was also a very large group of children that suffered from no teratogenic effects of LSD. So, as I said, it was difficult to really attribute these effects directly to the use of any psychedelic drugs. As it was demonstrated on animal models, which are, of course, for ethical reasons, more available for research, offspring born to females that abused psychedelic drugs during pregnancy may suffer from low body weight, skeletal defects, gait tissue, motor defects. There is also an increased mortality, number of stillbirths and miscarriages, and some teratogenic effects seems to be carried on onto the second generation. At present, there is a lot of interest in the zebrafish model of fetal development because, surprisingly, early stages of the zebrafish development resemble early stages of the human development during the first, second, and third trimester of pregnancy. But these studies are sort of only at a very preliminary stage at the moment. But they give a lot of hope that we might get a better insight into this area without the need to compromise the safety of women and children. In our research, we focused on two issues related to psychedelics and their structural analogs in the context of pregnancy. First of all, we wanted to predict the placenta permeability of those compounds by passive diffusion. And then we also wanted to study their interactions with placental enzymes responsible for detoxification of harmful xenobiotics. And we examined a large group of compounds synthesized by some researchers reported very recently in a number of publications. The compounds that we were mainly interested in belong to two groups: phenylethylamines, 130 compounds. These are compounds resembling mescaline. Ergolines, 9 compounds, which are structurally related to LSD, and tryptamines, 111 compounds, which are structural analogs of DMT, for example, psilocybin or psilocin. And ketamine, just as a sort of off-group compound of a similar activity. We wanted to study the ability of compounds to cross the placenta. Of course, to do so, we had to keep in mind that the placenta, which is an interface between the maternal and fetal compartments, is not a very tight, not a very absolute barrier. Its main function is to facilitate and regulate mother to fetus transport of oxygen and nutrients, and to remove the metabolites from the baby, unborn baby, to the maternal blood circulation. So obviously, the primary function, with the primary function of placenta being transport, it must not be 100% tight. Transport of molecules across the placenta, as I said, occurs mainly via passive diffusion, but there are also other mechanisms, like, for example, involving placenta transporters. And the whole issue is very important because pregnant women are exposed to a significant number of prescription and non-prescription medications, also to substances of abuse and to environmental contaminants, such as, for example, pesticides. In North America, for example, women use on average 2.3 prescription medication during pregnancy, but many consume 4 or more drugs. I mean, prescription drugs. And in addition to the different dietary supplements, over-the-counter drugs, and other substances. Coming from cosmetics, for example, all sorts of compounds that a user might not even be aware of actually being exposed to. In our study, we developed a novel model of the placenta permeability of organic compounds, with the placenta permeability expressed as logarithmic fetus to mother blood concentration ratio in the state of equilibrium. This was calculated according to an equation based on a set of 54 compounds with experimental log FM values taken from Takaku publications and from some other authors. And based on the calculated log FM values, we could assign our compounds to either the group of compounds crossing the placenta easily, PL1, or not crossing the placenta, PL2, and sort of an ambiguous group in between, with the cutoff values taken from Di Filippo's work published in 2021. The equation that we developed has quite good statistical parameters, and is based on 5 descriptors, primarily on Lipinski, which is a binary descriptor telling us if a molecule is drug-like according to the Lipinski's rule of 5. CACO2, which is basically a descriptor related to the permeability of compounds through CACO2 cells, human colon epithelial cancer cell line, used as a model of human intestinal absorption of drugs and other compounds. Topological polar surface area, and parallel artificial membrane permeability assay, which is also a screening method used to measure how well compounds can pass through an artificial lipid membrane. And the last but not least, a size-related descriptor, the total count of heavy atoms. And as we could see from the coefficient of the equation, all the parameters but one, all the parameters but the total count of heavy atoms have a positive influence on the permeability of compounds through the placenta. And the heavy atoms count, which is basically related to the molecule size, is negatively correlated with the compound's permeability. It was, according to our predictions, established that in fact all the psychedelics and their analogs, irrespectively of the chemical family, are likely to cross the placenta quite easily, with probably by the passive transport mechanism. So the next stage of our research focused mainly on whether these compounds interact or do not interact with the possible protective mechanism of the placenta. And as our target, we selected two enzymes, glutathione S-transferase GST and N-acetyltransferase NAT2, that are active in the placenta and protect the fetus from xenobiotics, unless, of course, they are inhibited by some compounds. And we estimated the affinity of a few parent psychedelics for these two enzymes using molecular docking. With glutathione as a reference compound for the GST enzyme and coenzyme A as the reference compound for the NAT2 enzyme. According to our estimations, LSD is the compound that is very likely to bind to both the enzyme quite strongly. Its affinity for both GST and NAT2 enzyme seems to be higher than that of the reference molecules and then of the other psychedelics that we consider at this stage of this study. We also studied or followed the possible structures of the enzyme ligand complexes, and basically what we concluded was that the amino acids that play the key role in the complex formation are leucine 35, leucine 119, and 115 for GST, and phenylalanine 93, phenylalanine 217, and valine 106 for NA2, respectively. With this in mind, we might expect that at least LSD-related psychedelics are likely to be strong inhibitors of the placental's protective system, and also we must be aware that all the studied psychedelics and their analogs are likely to cross the placenta, most probably by the passive diffusion mechanisms. Thank you very much for your attention.